Okay, hello everyone. So we're coming to the last few lectures in microbiology, and this lecture looks at viruses. So the different types of viruses, their structures, their life cycles, and their roles in human disease. Then in the next lecture, we'll look at viral diseases and the different antiviral drugs that treat them. So this lecture is important as pharmacists need to know about the different types of viruses, how they infect humans, how they reproduce, and how they're transmitted from person to person. So specifically, we will define what virology is, and we'll differentiate between different classes of viruses. We'll also cover the viral structure in depth, as well as the viral life cycles in bacteria and animals. We will then go on to cover the role of viruses in human disease and look at the difference between viroids, virusoids, and prions. So virology is the study of viruses and virus-like agents, including but not limited to their classification, their disease-producing properties, their cultivation and genetics. So mortality from viral epidemics has actually been dramatically reduced due to public health measures in the developed world. However, when epidemics occur, they can spread quickly with global air travel. For example, in 2009, there was an outbreak of flu across various continents, and obviously now we're seeing this with coronavirus. In addition, in early 2014, there were cases of Ebola in Guinea, which led to a massive epidemic in Western Africa, and Ebola is very deadly. So because of these kind of epidemics, this is why you have a lot of people working in, for example, the World Health Organization, trying to prevent these kind of outbreaks. So viruses are very small, generally smaller than bacteria. Bacteria are about 100 times larger. So unlike bacteria, we cannot see viruses with a light microscope, with the exception of some larger pox viruses. It wasn't until the electron microscope was invented in the 1930s that scientists could first see the structure of viruses. However, methods for the prevention and treatment of viral diseases occurred a long time before they were actually seen under the microscope. In fact, historical records say by 17th century, inoculation, also known as variolation, was used to prevent the viral disease smallpox in various parts of the world. So smallpox was a serious infectious disease caused by the variola virus, and this was fatal in 30% of cases. So the early symptoms are fever, fatigue, and severe back pain. Then a few days later, the virus produces a rash with bumps full of a clear liquid, which later fill with pus. These are called pustules. And these finally develop a crust that dries and falls off. The patients were then left with terrible permanent scars or pox marks. Variolation was a method of immunizing patients against smallpox by infecting them with substance from the pustules of patients with a mild form of the disease. This was in the hope that this would produce a mild protective infection and prevent a more serious, deadly case of the disease. Then, in the late 18th century, Edward Jenner realized that milkmaids who had caught cowpox from their cows did not show symptoms of smallpox after variolation. And Edward Jenner then started inoculating patients with cowpox to prevent smallpox. And this was the first vaccine to be developed against a contagious disease. In fact, the word vaccine comes from the Latin word vacca for cow. And smallpox has actually been eradicated since 1980 due to vaccination. So now the structure and genetics of viruses are well defined, but there continues to be new discoveries to reveal more complexities. And in humans, there are a wide variety of viruses which cause infections and disease, and these range from the very mild, so things like colds, to the very serious, such as Ebola and HIV, and not all of these infections have treatments. In fact, some of the deadliest emerging pathogens in humans are viruses, but there are few treatments to actually cure these diseases. By emerging pathogens, we mean pathogens that have newly appeared in a population or have existed but are rapidly increasing in incidence or geographic range. So the continuing emergence and re-emergence of novel and dangerous pathogens with the potential to cause epidemics is a permanent threat to the health of populations around the world. So viruses were first discovered after the development of a filtering device which could remove bacteria from any liquid sample. So in the late 19th century, it was found that a disease of tobacco plants, this is called the tobacco mosaic disease, or TMD, it was found that this disease could be transferred from a diseased plant to a healthy one via liquid plant extracts. Then in 1892, Ivanovsky showed that this disease could be transmitted even after the filtering device had removed all viable bacteria from the extract. Ivanovsky therefore concluded that the cause of TMD must be an extremely small bacterium or a bacterial spore. However, it was many years before it was proven that these filterable infectious agents were not simply extremely small bacteria, but a new type of tiny disease-causing particle. And then it was Beijerinck in 1890 who concluded that it was possibly a chemical, like a biological poison. So the word virus is actually Greek, meaning poison. However, it is Ivanovsky who is credited as the original discoverer of viruses and a founder of the field of virology. So viruses are a group of infectious agents that are unique in their simple acellular organization and in their pattern of multiplication. So acellular means that they don't have cells, and therefore they have no cytoplasm or cell organelles. Their acellular nature means that they are not included in the tree of life. The tree of life is used to explain the relationship between the different species on Earth. The tree of life consists of 3 domains: archaea, bacteria, and eukarya. Viruses can exist extracellularly or intracellularly. When extracellular, they are inactive because they possess very few enzymes and they cannot reproduce outside living cells. Therefore, viruses are obligate intracellular parasites, as they must infect a host to survive and reproduce. When intracellular, viruses exist mainly as nucleic acids that can take control over host cells and use them to synthesize viral components, from which new viral particles, called virions, are assembled and released. The new virions then can transport the viral genome to another host cell to carry out another round of infection. So next we're going to look at the characteristics of viruses. As we said, viruses are infectious acellular pathogens. They are obligate intracellular parasites that infect specific hosts and cell types. They contain a DNA or RNA genome, but not both. The genome is surrounded by a protein capsid, which is a protein coat, and in some cases a phospholipid membrane studded with viral glycoproteins. Those covered with a phospholipid membrane are called enveloped viruses. So viruses lack genes for many products needed for reproduction, so they require exploitation of host cell genomes to reproduce. So viruses can infect every type of host cell, including plants, animals, fungi, protists, bacteria, and archaea. However, most viruses will only be able to infect specific cells of one or a few species of organism. This is called the host range. All viruses must attach to a host cell long enough to gain entry into the cell. Attachment to the host is accomplished by specific interactions between molecules on the surface of the virion, which is the virus particle, and the receptors on the host cell. So receptor specificity is at least partly responsible for the preferences viruses have for a particular host. For example, bacteriophages are viruses that infect bacteria, but these often infect only certain strains within a given species, so they're very specific. Likewise, animal viruses infect specific animals, and in some cases only particular tissues within that host. However, if the receptor recognized by a virus is present in numerous animals, then the virus will infect more than one animal species, and such is the case with the rabies viruses. So the host range will depend on the presence of receptors for the virus on the host cells. If a host cell lacks the receptor for a virus, or if the host cell lacks some component necessary for the replication of a virus, the host will be inherently resistant to that virus. For example, mice lack the receptors for polioviruses and thus they are resistant to poliovirus. Similarly, humans are inherently resistant to plant and many animal viruses. So viruses can be transmitted through direct contact or through indirect contact with fomites. So the definition of a fomite is an object or material that is likely to carry infection, so things like clothes, utensils and furniture. Or viruses can be transmitted through a vector, which is an animal that transmits a pathogen from one host to another. Arthropods, for example, mosquitoes, ticks and flies, are typical vectors for viruses and can act as mechanical vectors or biological vectors. So what do we mean by that? So mechanical transmissions occurs when vector carries a virus on the outside of its body and then transmits it to a new host by physical contact. Whereas biological transmission occurs when the arthropod carries the viral pathogen inside its body and transmits it to the new host by biting. Zoonoses are viruses that can be transmitted from an animal host to a human host. For example, avian flu originates in birds but can cause disease in humans. Then you have the opposite, so reverse zoonoses caused by infection of an animal by a virus that originated in a human. So examples of these are methicillin-resistant Staphylococcus aureus, known as MRSA, and influenza A virus. So as we said in the introduction, generally virions, the virus particles, are small and cannot be observed using a regular light microscope. They are much smaller than prokaryotes such as bacteria and eukaryotes, which is actually an adaptation which allows viruses to infect these larger cells. So virus size ranges from very small 20 nanometers up to 900 nanometers for some larger viruses. As you can see on the diagram below, the poliovirus is very small and can only be seen with an electron microscope, whereas the smallpox virus is much larger and can be visible under a light microscope. Recently, new giant viral species have been discovered, such as Pandoravirus salinus and Pithavirus subiricum, with sizes similar to a bacteria cell.